This is a DNA replication fork. The origin of replication is here, 5'. It goes 5' to 3', and it moves forward, opening like a zipper. Now the backbone here, represented by the blue tape, is made of phosphodiester bonds, which connect the 5' phosphate group to the 3' hydroxy group. Our first enzyme here, represented by the beer, is gyrase, which is responsible for relieving supercoiling ahead of the replication fork, and it does so by nicking the DNA backbone to release tension. Next here is helicase. Helicase is an enzyme that attaches at the replication fork and moves the fork forward by breaking the hydrobonds, thereby separating the DNA into two separate strands that can now be replicated. Okay, now my K-cups are the single-stranded binding proteins, and these proteins work to keep the strands from coming back together near the replication fork, which result in double-stranded DNA called hairpins. Okay, so now we have this glass here. This glass here is primase, which is essential for making RNA primers, the marbles. And RNA primers are very important because they provide our DNA polymerases a 3' hydroxy group to add their nucleotides to. Without primase, there would be no RNA primers, and our DNA polymerases would not be able to replicate. Okay, now DNA polymerase 1 here is the green way, and DNA polymerase 3 is shown by the purple way. Both enzymes catalyze DNA replication and can add nucleotides in the 5' to 3' direction. In addition, the DNA polymerases have proofreading powers because they can cleave nucleotides in the 3' to 5' direction, those that are not complementary bases. DNA polymerase 1 is also important for removing RNA primers and replacing them with new DNA. However, it can only work in small fragments before they fall off. DNA polymerase 3, on the other hand, can work continuously due to a clamp that keeps the strands together. Thus, we have a leading strand and the lagging strand. On the lagging strand, DNA polymerase 1 has to work ahead and then jump back in order to work 5' to 3', because if it were to just go this way, it would be replicating 3' to 5', which is not how DNA replication works. So because it has to do this jump ahead, come back thing, this results in these fragments called Okazaki fragments. Lastly, we have ligase, which acts as a glue. So ligase here is going to be used to seal these Okazaki fragments, and therefore they can be joined together to form one continuous strand. So this side would be the leading strand because DNA polymerase 3 is working continuously and there are no fragments. However, all of these enzymes are used to correctly and accurately replicate DNA. Without these enzymes, we would most likely have either no DNA replication or a lot of mutations.